Okay, hi, good morning. If you can see us, we are live from the lab. So here, this is Dr. Khatijah.
Hi, I'm Dr. Khatijah.
So today we're together with you, Dr. Khatijah and I, and we have Puan Azima, the medical lab technologist. So we will share with you, we will do a demo with you about how to do a blood smear. All right, can you hear me clearly?
Yes,
Yes,
doctor.
doctor.
Yes.
Yes,
Yes,
doctor.
doctor.
We will demo with you on how to do a blood smear or blood film, thick and thin blood smear, especially for malaria parasites. So usually in the hospitals or in the health facilities, that we write BFMP. BFMP means blood film for malaria parasites. So today we are going to show you how to do a finger prick. Okay, the demo will be done by both Dr. Khatijah and Madam Azima. All right, and then we will show you how to actually do the staining and so on. Okay, but before this, right, I want to show you the laboratory that you are really miss so much. Okay, since you haven't come to this lab yet, so let me change the camera. All right. So these are the camera. This is the laboratory that you really miss. All right. Right. So here is a support where you are supposed to sit. You have one big desk that can fit more than 10, I think, or 10, exactly 10, maximum of 10. So you can see even one to be a group can sit together in this one bench. Right. It's a very huge lab. You see here, there's Dr. Khatijah and Madam Azima. Right. So hopefully when you come back in year two, we really want you to come back when you enter your year two at least. That okay, this is Mr. Azhar, he's taking my images. So Mr. Azhar himself also a medical lab technologist, all right? So he is also helping us in this lab practical. Okay, good. Right, so let me stop sharing this one for a while and do some setting up. All right. So for the preparations of blood smear, so basically what you need, of course, you need a microscope slides. All right. This microscope slides, I think you have seen microscope slides before, right? It has to be a clean one. So before you do the slides, the blood smear, you need to check whether they are really clean, very clean, or if it's not clean, you need to wipe it, wipe it with ethanol or gauze, or as long as it's very clean. Okay, or otherwise you can just wipe it uh, clean like this. Okay, then you need alcohol swab. Okay, the functions of alcohol swab is actually to disinfect the skins, to sanitize the skins from any pathogens or skin bacteria and so on. All right, so this is an alcohol swab. So you also need a clean cotton wool or cotton swab. Okay, once you have prick, you will remove the excess of the alcohol or the blood. Okay, and of course you need a lancet. This is an automatic lancet. You can just press and we just prick your finger and the blood will come out from your skin. Okay, and then right. So not only that, you also need a spreader. A spreader is basically is another glass microscope slide. It's actually for you to do the smear, to spread for you to do the thin blood film. Okay, so this is called a spreader. And then there is a for finger prick for pressure on the blood smear. Okay, then for staining, this is what we use. This is a Giemsa stain. Okay, I don't know whether you can see this clearly. This is a Giemsa stain. So this is the what we use, and its color is very dark purple color, right? And of course, in order for us to prepare this Giemsa stain, it need to be diluted with a buffer, okay, and a buffer with a pH 7.2. And to prepare the buffer, we use what we call by GER buffer, GER tablet. And then it has a pH 7.2 here. Okay. But then in the laboratory, we usually prepare it in what we call a Coplin jar. In this Coplin jar, we have a 3% Giemsa that's already made, right? And of course, we also have the 10% Giemsa in this Coplin jar for staining, right? And very important, methanol. Methanol, if you read at the guidebook, the laboratory practical guidebook that I shared with you, methanol is for you to fix the thin film, right? Okay, let's start with this. So you can see, so in the process of preparations of the blood smear, okay, if you look at the camera here, right? Okay. So, Madam Azima, we do a finger prick on Dr. Khadija's finger. Right. So first, she will. Can you see it clearly, right? Is it clear, yeah, from your screen?
Yes, doctor.
Yes.
So Dr. Khadija will do
Volunteer. Okay.
voluntary prick by Madam Azima. So Madam Azima with the finger is clean first using alcohol swab. Okay. And there's a lancet. So basically, she will press the fingers so that there will be a lot of blood on that side. Right, you can see the blood there. So first, you need to wipe the first blood there. Oh, it's very. Seems that the Khadija has lots of blood. So this is how you prepare the blood smear. So lots of blood. This is for the thin smear. For the this is lots of blood. For thick smear, usually we have a few drops. You can do more than one slide. Actually, you can wipe first. Do more than one slide. Okay, this is how you do it again. This is for thin smear, only one drops. Thick smear, you can use 3 drops. 3 or 2 drops. It depends on the amount of blood. Since Dr. Khadija's blood has like too much, 2 is enough. Okay, then you wipe and clean. Thank you, Dr. Khadija. Okay. So, okay, for the preparations of a thin smear, okay, this is how actually you hold it. Yep. Slowly. And then you spread it out easily. Okay. So this is a thin smear. Then you can try again. This is quite a very thick thin smear because Dr. Khadija blood like too much. right? Then you spread it slowly. Yeah, so this is actually a very good thin smear. That's how you do a thin smear. Okay, then you just let it dry. For the thick smear, you use at the edge of the spread, angle of the spreader, you just make a round circle. Okay. Bigger round is better actually. If you can do a bigger circle, then you can spread the thick blood film. Because if the thick blood film is too thick, okay, it takes longer time for it to dry. So it is advised you make a very big circle, right? Right. Okay. So now here, usually we just let it dry. If you look at this thick blood film is so thick, right? So it takes such a longer time to dry. For the thin film, because it's very thin, it's quite fast for it to dry. So sometimes in the lab, for us to dry, especially the thick blood film, okay, sometimes we can just use a hair dryer. We just dry it off. Okay, so we dry. So make sure the thick blood, especially the thick blood film lah, it needs to be dry, really, really dry, because otherwise, when you want to stain it, if you don't dry properly, when you want to stain it, it will scrape off from the slides. Okay, so here we can just try for a while. So let me wear my glove. Okay. So far, is it clear, everyone?
Yeah.
Yes,
Clear.
doctor.
Yes, doctor.
Yes, doctor.
Yes.
So now I just put this one aside. This is just like a cooking channel lah. So you already prepare you already prepare the one that is already dry. So this is how it looks like when it's really, really dry. So very nicely dry, and then even the thin film is very, very thin. Okay. Sometimes in the hospitals or in the field, they usually prepare a thick and a thin film in one slide. Okay, thick and thin film in one slide, because in the hospital they want to be fast. You just drop the blood there, right? But it is advised not to have thick and thin blood films in one slide lah. So let's say in the future, you become a doctor in the field or something like that, if you want to prepare slides, so it's advised to have a separate thick and thin blood film. Okay, all right. This one we can put it aside to let it dry by itself. Right. So what are we going to do with the thick and the thin blood films? Right. Okay. This is a methanol. So remember, only for the thin blood film that we fix it with methanol, not the thick blood film. Right. The functions of the methanol is actually to let the blood film stick on the glass, and then it will not rupture. It prevent the red blood cells from rupture. Right. So how do we do that? Okay. It's very easy. This is on the coffee jar. This is methanol. Okay. You just dip it for a few seconds. Let's say usually we dip it for 5 seconds is enough. So that is. So you just count 1, 2, 3, 4, 5. Okay. Then you just let it dry. Okay. So methanol, because it's a type of alcohol, it dries very fast. Right? Just let it dry. Either you can try it on the glass slide rack. For the thick blood film, remember we don't fix it, because the reason why is because we want to let the red blood cell rupture. Once the red blood cells rupture, you can see the parasite clearly. You can detect the parasites. You can do the counting of the parasites on the thick blood film. On the thin blood film, we want to retain the morphology of the red blood cell, the infected red blood cell, the parasites. So that's why we fix the thin blood film. Right? You just let it dry for a while. Actually, it's dry quite fast lah. Yeah, I think it should be fine. So, if you refer to our guidebook, the laboratory guidebook, there's a different percentage of GIMSA for different slides, but for different types of blood smear. Okay. For the thick blood smear, all right, you can see for the thick blood smear is you can either use a 3% or a 10%, but it depends on the type. If you use a 3%, so you have to stand slightly longer. If you use the 10%, okay, you can stand it for 30 minutes. But in the hospital, sometimes the doctor wants the results quite fast. So that's why in the hospital, they just use one type of GIMSA, which is 10% GIMSA, right? But in order to have a very clear image of the parasites, so it's better to stand it longer. Okay. So here, I will just stick to the 10% first lah, because we need to do it very fast. So this is 10%. So for the preparations for staining, this is an applicator stick. Applicator stick, you just patah it macam lidi lah. Okay, you put on the container like this. Okay. And then for the slides, we just usually this is the top, this is the bottom. So you just invert it, facing down, arrange it like this. Same goes for the thin film that's already been fixed. Right? So you arrange it nicely like this. Cantik-cantik. Okay. Sorry lah, singit-singit sedikit. Perfectionist, so have to be lurus. Okay. Now everything is facing down. So using this GIMSA, okay. So this is how we actually proper. You have to mix it well. This is how you actually stain it. Kat bawah, you drop it at the bottom. So we spread to the smear. So this is a thin film. For the thick film, the same thing. Okay. So very nice, very clear, very clean. So this one you can stain for half an hour. Just leave it for half an hour, right? And then another alternative for staining, sometimes in the hospital also, if you want to be very fast, you can also dip it inside this Coplin jar. Inside this Coplin jar. Okay. For dipping, you can dip like many slides at one time. I think 5 slides is it at one time? Yeah, 5 slides at one time. Okay. So now we don't have to wait for 30 minutes, but now actually just want to show you this how actually we need to stain the slides. It's inverted, and then you using a plastic pressure pipette, and then you just apply the GIMSA at the bottom of the slides, and then you just go there. This is a very clean way, right? You can see it's not that messy. Okay. Right. So any questions so far? Is everything clear?
Doctor, blood smear just now. Okay.
Yes.
So we are going to use one of the edges of the slide to
Yeah.
make a round shape. Okay.
Yeah.
So after we done with one blood smear, one tick, should we change the edge for another?
Uh, no need actually. You can just wipe it with your clean it like this, with your clean cotton wool. Then you can continue doing it using the same spreader. You are referring to the spreader, kan?
Yeah, correct.
So no need. This spreader is just like to spread lah. You don't have to be. It will not contaminate the other slides. So you just have to clean it nice. Just clean it nicely. You can clean it with alcohol. You clean it nicely, then let it dry. Then you can continue with the other slides or other patient, for example. Is that clear? Did I answer you?
All right, clear, doctor. Thank you.
Any other questions? While waiting for this one to stand for a while, just spend a few minutes lah.
Doctor,
Yes.
is there a difference if we stain it like not inverted?
Actually, there's no difference. But if you want, usually when we apply the, here in UNIMAS, we have tested a different way of staining. Kan? Usually for this Coplin jar method, okay, this one, the GIMSA, is being recycled for many times, right? So after you in the hospital, in the hospital they have to be very fast, many samples kan. So they will put many slides at one time. After that, take the slice out, okay, just leave it there, and then the new batch coming using the same GIMSA, masukkan balik semula the slide inside here, okay. So meaning to say you recycle this for how long we don't know. Usually they will change by daily basis, right? But then this will produce debris or some dirt from the slides, and making the GIMSAs not clean, not clear. While this method, the one that I showed to you now, is a very clean method because you're applying a fresh, clean GIMSA on the slide. Right? You can also you can also stain it when on your slides upright kan di atas. But then if you apply it like this, drop it like this, it will move lah, it will go here and there lah, it will not stick there lah. But if you inverted it like this, you can see the GIMSA is not moving anywhere. It stick there.
It's me immerse perfect.
The smear actually immersed very perfectly, very nice, cantik. And then the result from this kind of method, it will give you a very clean blood film. No debris from the GIMSA lah. Is that clear?
Yes, doctor. Thank you.
Okay. Any other questions? Are we waiting for this one? So which blood film that we need to fix with methanol?
In the
Thin blood film.
Blood film. So what happens if you fix thick blood film?
Red blood cell will not rupture.
Yeah, red blood cell will not rupture kan. And then what happens if the red blood cell, when the red blood cell not rupture and then it will kumpul on the slide, right? Clump like that on the slide. And then you look under the microscope. What do you think will happen?
Cannot identify the parasite.
Well, not really. You cannot identify the parasites. This is if it's already like it is fixed with methanol. The red blood cell is not ruptured. Kan? When you look, you know how to use a microscope. You used a microscope before, kan? Right?
Yes.
Yeah. So the lights kan datang dari bawah. So when it's not ruptured, the light cannot pass through the slides because it is covered by the unruptured red blood cell. So you cannot see anything actually. Dia kena halang lah. But if you rupture the red blood cell, the light can pass through when microscope dari bawah, dari atas, you can see clearly, clear background, but you can see the parasites on the slides under the microscope. All right. Okay. Still while waiting, which blood smear that we use to count parastemia? Thick or thin? Anybody want to guess?
Thick.
Sorry? Come again?
Thick blood smear.
Yes, thick blood smear. Because for the thick blood smear, you can find a lot of parasite in one pill that you can count it easily. Thin blood smear, because it's very thin, it's spread out. You have to spend so much time to look for the parasite for you to count. That's why we use thick blood film for estimating the parastemia to count the parasite. Okay, I think imagine that we already half an hour. Just want to cut short time. Imagine that we already have passed through half an hour, so this already stayed nicely. So we can do step. Can take this out. Okay, we can see it's already like stained. Okay, it's already stained nicely. Right? For the thick film, you can see it stained nicely. Can you see it clearly? Nampak?
Yes, doctor.
Yes, that's right.
Okay. How do we want to wash this? Is another skill to wash away the gimsa. Okay. So, Madam Azima, can you help me? So, Madam Azima will show you on how to okay, and I will show you the. Okay. Right. So this is how you actually wash the gimsa. Okay. Your water need to be very, very slow so that it will not disturb the gimsa. Okay. You can see it's already washed. Okay. Right. Then you can just let it dry. Just let it dry, or you can just use a hair dryer. Okay, you can just clean it and it has a very nice smear, and it can be should be look like this lah. Once it's dry, it's very nice thick and thin film of using the Gimsa stain. Okay, is that clear so far?
Yes.
Okay. Going back to this slide, you can see this is the thick film from Dr. Khadija's blood. You can see it's very thick, so it takes such a longer time for it to dry. Okay. So I do not know how it can be. This one, the thin film. The thin film is already dry. It's very nice and dry. Okay, and this one we can fix with it with methanol. All right. So when it is done, okay, once the blood film is done, you can actually look under the microscope. Okay. 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 For the microscope, usually when you put it on the platform here, usually you will start with a low magnification first. You go for the low magnifications, then from the very low to the next one that you go up and on the thick that film lah, and then to 40. Usually 40 we use Ada 40. It depends on if you have 40 or 60 or 100 of the magnification here. You can see here 40. Okay. And then once you can identify, once you detect the parasites, right? So that's where you use here 100 magnifications with immersion oil. For immersion oil, usually for you, if you want to detect, you want to identify the species using the thin blood film. Okay. For counting of parasitemia, we don't use a very high magnification. For counting of parasitemia, it's okay to use 40 times for counting of parasitemia. So, for counting of for the parasitemia, okay, we usually need this is a counter. So when you look at the microscope, you just click. Once you find, you have the other type of counter. This is a so this is not a proper counter that we use now. We usually use a this type of counter in the lab to do differential count. Macam we count berapa trophozoite, how many trophozoite, how many schizons, how many gametocytes, and so on. So you can sometimes in the lab, the sounds usually beating like this, and also usually for the end part, we save it for we count the white blood cell. So usually here trophozoite, schizons, gametocyte, and end here, or the fourth one will be the white blood cell. So this is how we usually count. So usually in the lab we sound bunyi like this. If you hear this sound, that means people are busy doing parasite count. Not only the hematology counting of the different the white blood cells. Okay, is that clear so far?
Yes,
Yes.
doctor.
Yes, doctor.
My stomach is so big. So, okay, so now I'm going to share with you what happened. I think I'm going to stop sharing this one first. So I'm going to share with you the different slides. What happened if we fix the thick blood film? Okay, so I'll show you an example. Right, can you see this?
Yes.
Yes.
So this happens when you fix the thick blood film. You can see the red blood cell, they are actually not lice here, but they are all clump. You can see the thin. You can see the thin. You can see the white blood cell, right? The white blood cell in the purple color, but you can see the red blood cell. They are all clumped together, and then you cannot identify the parasites from here. Actually, you can't even identify the red blood cells, the structure of the red blood cell. All right. Then let me close this one. If you unfix, if you don't fix the thin film, okay, here, what happened if you do not fix your thin film? You can see basically the red blood cell is not like a proper a common red blood cell. You cannot see the concave, the color of it. You can see a very pale. This is actually the red blood cell is already ruptured. It doesn't look like a red blood cell anymore. Okay, but you can see these white blood cells. The white blood cell is still clear lah. Okay, so this is a bonus question. What type of white blood cell is this? Have you learned hematology?
Hypersegmented neutrophil.
Neutrophil, pandai. Okay, good. So you can identify different types of white blood cell. This is a neutrophil. There are five lobes here. Okay, then yeah. So you can see this is what happens if you don't fix your thin film. Right. So let me unshare this. And an example, I'm going to show you the example of a thick blood film. Okay, can you see this?
Yes.
Yes.
So this is a good thick blood films. All the white red blood cells are all ruptured, and you can see the parasites here. Right? You can see the chromatin dots. You can see the structure, the cytoplasm of the parasites very clearly. So that's the reason why you don't fix the thick blood film. You want the red blood cell to rupture so that the parasites can be seen clearly. Okay, a bonus question. Can you identify this species of Plasmodium? Anybody want to guess?
Maybe Plasmodium.
Yeah, it's Plasmodium, but what species? Can you try? Do you want to guess what species is this? What species of Plasmodium? Is it Falciparum, Vivax, or Nosai?
Plasmodium vivax.
Yeah, because see that's the name of my slide is Plasmodium Vivax kan. It's very obvious. Right? So basically this is Plasmodium vivax. Because for Plasmodium falciparum, you can only see the chromatin dot and the ring form. And here you can see it's a very huge, large parasites, very clear parasites here. So it's a Plasmodium vivax. Okay, so let me stop sharing there and open you the how does a falciparum look like. Okay, so a falciparum, this is how it is look like in the thick blood film. You can see the chromatin dot and the ring form, and this is a very typical characteristic of Plasmodium falciparum thick blood film. Okay, then and I showed you thin blood films of Plasmodium nosai. Okay, so this was, if you can see this, this was taken from patients admitted in Kapit Hospital. All right, you can see a very nice. This is the method that we use, the staining method that we use. That we went to Kapit Hospital in Sarawak, and we teach them on a proper way of staining it so that we have a very clear image under the microscope. No debris, very clear red, right? And also you can see the image of the parasites using the method that I showed you just now on how you stain it. You invert the slides, and you use a fresh gypsa stain. Okay, you can see the structure of these parasites of Plasmodium nosai. And bonus questions. What structure is this?
Schizon.
Yeah, so it's actually a schizons. You can see, you can count how many merozoites inside here. The nucleus here are representing the merozoites. So usually under the microscope, if you want to search for a parasite, there's a systematic way. You just don't put the slides under the microscope and you just find it randomly, kiri, kanan, depan, belakang, you move the field. Usually you start at one edge. from top to the bottom, and then once you reach the bottom, then you move your field to the right or to the left. If you go to the left, you go to the left. Then from the bottom, you naik atas, search for the parasites, right? And go to the top. Then you move the field again to the right. Okay. Then you go down and go to right again. You go up, right, down, right, up. This is a systematic way for you to search the parasites in the thin blood film, or even to count parasitemia in the thick blood film. If you go by random, you might not know whether you have seen that spill or not. Okay, so this is a very important about how you want to identify or how to want to do microscopy examinations of the thick and the thin blood film. Okay, for the thick and the thin blood film, it's not only useful for the malaria parasites, but in Malaysia also we can use it to detect other parasites like filariasis as well. Filariasis, we usually use the thick blood film because once you rupture the red blood cell, you can see the microfilariae very clearly. All right. Okay, I think I've covered everything. So, any questions so far with the with this demo just now? Any questions?
No.
No, doctor.
No.
Okay. So I guess everything is very clear. Okay, with you. If there's no questions, okay, as usual, I want to show you this one. So I actually need your feedback. Okay, so if you go to www.menti.com and apply this code, all right. So in this COVID-19 pandemic situation, what do you think about this live parasitology practical session that you have seen just now, showing you about how to do the blood smear? So you can apply this and you can give your answer freely. Okay, while waiting for that one, right? So while waiting for this, you give a few minutes lah. If you can go to www.menti.com and you apply the code 51484144. Can you see it? Can you see this? Is it showing?
Just now only white.
Neman, tutup balik. Show balik. Okay. So your feedback is very useful. So I don't know if any of other lecturer have done this live practical sessions about the demo. Okay. So the reason why I'm doing this is that in reality, when you are in this laboratory, okay, you will actually do this blood finger prick on your friends by PayLah, or you cucuk your friends. Okay, you do your blood smear and you stain it, and your friends balas dendam lah, you cucuk again your other friends. Okay, and you prepare the blood smear and you do the staining of the blood smear yourself. Right. This, and also at the same time, so you're also doing the practical. You also had the opportunity to actually to look at the parasites itself while waiting for the blood smear to dry, or for it to actually stain properly, and you have the opportunity to look under the microscope yourself on how does it look like if you fix your thick blood film, and how does it look like when you don't fix your thin blood film. But because of this situation, everything is online. So I'm sorry that you really had to miss that. So that's why I grabbed this opportunity for you to bring to the lab. Okay, so we have a few feedbacks here. Okay, very interesting. I miss doing lab practical. So everybody is missing doing missing lab practical. We also missing the student coming back here to have to be with you in the lab, because the lab has been sunyi for so long since last year. Right? It's amazing, best, tapi still nak balik campus. Yeah, we really miss you. Come back here. It's so cool watching it live, making me like we ourselves are in the lab. Okay, it's fun and make me want to try it myself. You can try it at home. But you can buy the lens set. You can buy the lens set in the pharmacy, but you need to be clean lah. But you need to have your own microscope and the Jimsa stand. Right, very good. It's helped me to understand better as I can see the method done live. Okay, it's immersive, excited. Immersive is very useful. At least we can see how it is done in real laboratory and ask questions along the way. Fun and exciting. It's great. More understandable. I wish all of the lecturers done this as well. Thank you for spending time with us. You're welcome. Effective, save time, and communicative. Because the reason why I'm doing live, because I want questions from you if you don't understand. So that's why it's very important that I usually have the opportunity, give it to you to ask a lot of questions if you don't understand. Okay. The practical session is fun and exciting. This one of having hands experience, I'm able to learn the technique and skill. Okay, it's very nice. I want to come to UNIMAS. Yes, we want you all to come here and experience the university life here. Easy to understand. Best sangat, they should be practice more. Just that you, hopefully, you will come back here for your year 2 next year. Is it next year? End of this year, right? But if the pandemic situation occurs, we have to do this live session like this again for the next year punya parasitology practical. Interesting. Miss Dr. Tu. Very good. You are the first conduct this excellent session. Dr. Paul, thank you, whoever you are. Okay, it was so good. The lecture explained very well. Excited. Okay, very clear demonstration. Very helpful. Quite a good comment. Thank you so much. I really appreciate your comments because if you really appreciate these live sessions, so perhaps we can introduce this to other lecturer of other discipline as well for them to do so. But of course, it depends on them lah. Right? More understandable. You can ask question, interaction. Yes. Practical like this is fun, but we appreciate our lecturers effort on doing a virtual lab practical. Yes, thank you, doctor. Thank you so much to you too. Yeah. Oh yeah, aja. Enjoy great experience demo. Best way to practice. Thank you, doctor. If we go back, do we have the chance to do this procedure? Yes. Maybe you're gonna have the chance to do this procedure. There will be chances for you if you come back here because we have to alter our way of teaching. Since online you cannot do this, but when you come back here, we try to put some slots for you to do this yourself. And opportunity for you to torture your friends to chuchuk their fingers. Okay. Interesting. We missed his live sessions. Is Dr. Khatijah okay? Yeah, she's fine. Although she bleeds a lot. And the thick blood smear is drying. Yeah, she's okay. I don't know why she's like bleeding so much. Okay. Right, thank you. Thanks, Dr. Khatijah. She says thank you. Okay, if there's no more, let me unshare this. Thank you so much for your comment here. Ada lagi? Tidak ada. Oh, ada. Terbaik. Terbaik. Tidak ada. I think I stopped sharing this one. And then I will share with you the attendance. If you have not scanned your QR code. Okay, so I think we have finished with this live demo of the practical sessions. The video clips I have pasted it in the e-lib for you to look again on the how to do the blood smear, thick and thin blood smear. You can find it in e-lib as well. That's been recorded by Madam Azimah herself and also Mr. Azhar. All right. Okay. Any question before we end these sessions? Final questions. Since your next week is your exam kan, EOB. No questions?
No, doctor.
No, doctor.
Okay. So I hope that everything is clear. And I would like to take this opportunity for you for your annual block next week. Hopefully you can answer. There will be, I think there will be 2 OSPE for parasitology. And usually, the OSPE will have related to the lab practical, the identifications of the parasites, and then you answer the question based on the parasites, based on the identifications of the parasites. That is what in the OSPE for the parasitology lah. Okay, and it's not that difficult if you really understand all the parasites that we have taught you lah, right? And also, do not forget to go to e-lib for the lab practical assessment. You can refer to your notes while answering the question. I have no problem with that, right? But it's important that all of you to participate in the assessment because it will contribute to the continuous assessment. Okay, so if we have no more questions, okay, I think we can end this session. So I will just let this QR attendance open for others to just continue scan for the attendance. Okay, so thank you so much for your attentions and for your participations in this practical. I wish you all the best in your EOB next week. Okay, thank you.
Thank you, doctor.
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 Thank you, doctor.